Hello everyone, my name is Simone Rocchetto. I'm a gastroenterology resident at the University of Milan, where I complete my endoscopic training at Valduce Hospital in Como. I'm really excited to share with you a paper we recently published titled Added Value of a Balloon-Based Mucosal Exposure Device in Computer-Aided Detection Assisted Colonoscopy, a randomized controlled trial. So why did we do this study? What motivated us? We live in an era where artificial intelligence is increasingly integrated into medical practice. So our curiosity was to explore whether combining a mucosal exposure device, specifically at balloons that flattens colonic fold, with computer-aided detection could further improve neoplasia detection. While previously studies have demonstrated the benefit of other exposure devices like the endocuff, there was no evidence on the effect of GI balloon associated with CADe. Personally, I was driven by the idea of contributing to innovation in endoscopy, evaluating whether a relatively new technology could meaningfully enhance the detection of precancerous lesions and ultimately impact colorectal cancer prevention. So let's talk about the results. We enrolled over 600 FIT positive patients from a colorectal cancer screening program. Participants were randomized to undergo colonoscopy with CADe alone or with CADe plus the balloon-based mucosal exposure device. But really importantly, our primary outcome was not a conventional adenoma detection rate, but a more clinically relevant endpoint, the detection of high-risk findings based on the ESGE guidelines, such as multiple low-risk adenomas, advanced adenomas, or advanced serrated lesions. For sure, this makes our study design unique and aligned with real-world clinical priorities. But what did we find? No significant difference, neither in the primary outcome nor in the secondary ones like ADR, advanced ADR, PDR, or SSLDR. Although the combined strategy did not result in improved detection rates compared to CADe alone, this finding still remained valuable. In fact, it helps us better define the context in which technological enhancements may or may not translate into meaningfully clinical benefit. But rather than pointing to the limitation of the exposure device itself, these results likely reflect the already elevated baseline performance achieved with CADe. In this setting, the benefit of additional adjunctive tools may be reduced by a ceiling effect. So what's next? First point, while artificial intelligence has shown promise in clinical trials, we must now focus on its actual impact in a day-to-day practice, where benefits may be more modest and uncertainty for sure still remain. Second point, our finding raised important questions about the role of mucosal exposure device in daily endoscopic practice when artificial intelligence is integrated in colonoscopy practice. In fact, despite promising theoretical advantages, their use remained limited in clinical practice. So in conclusion, this was the first randomized control trial to evaluate whether the combination of a balloon-based mucosal exposure device and artificial intelligence in colonoscopy. Although the combination did not improve detection rates compared to CADe alone, our results contribute to refining the role of emerging technologies in modern endoscopy.